टाइम लगेगा रिकॉर्डिंग होने को हो गई. So very good evening to one and all. Today we have with us Dr. Shammy Jindal. He is working as associate professor in Laurate Institute of Pharmacy, Kathog, Himachal Pradesh. Dr. Shammy is having 14 years of experience in teaching and research, and Dr. Shammy Jindal have published near about 40 review and research paper. Currently, he is guiding around 16 MPharm student and one PhD student under his guidance. And he is the editor of various journal of repute, for example, in elsewhere, Springer, Taylor, and Francis. And Dr. Shammy Jindal have organized multiple conferences, workshop, and FTP program under his leadership. So I welcome you, sir, in the ISFCP dialogue series for your lecture.
Thank you, Dr. Amish Sharma, sir, for the kind introduction. I am filled with profound gratitude and excitement as I stand before you all today. It is truly an honor to be invited by Director cum Principal Professor Dr. G.D. Gupta, sir, who has been my mentor and guide throughout this journey. I owe a great debt of thanks to him for his an invaluable guidance and mentorship, which has brought me to this moment. So, don't spending much time. I'm going to start my presentation. The today's our topic is uh, drug discovery. So, I'm going to share my presentations.
Yes, sure, sir. I will let you know when it is visible.
Okay.
यस सर दिख रही है ठीक है फुल
Sir,
स्क्रीन कर लो
visible?
यस फुल स्क्रीन कर लो बस
कर दिया सर फुल स्क्रीन.
ठीक है सर विजिबल है ओके
Okay, so today's our topic is that is a drug discovery and development. Myself, Dr. Shammy Jindal, Associate Professor from Laurate Institute of Pharmacy. So this is the drug discovery is the most prominent area and main area of our pharmaceutical field because all we are working on our drugs and the drugs are to be discovered by our pharmacists, doctors, fellow researchers. So, if we will talk about regarding the drug discovery, in the past, most of the drugs are discovered by from the traditional remedies or serendipitous discovery. But nowadays, we know the disease at molecular level, physiological level, as well as we know the shape of the molecule at the atomic level. So nowadays drug discovery becomes a vast area where we can do many things, many things, and it is a short way how we can discover a drug for a new disease. So it is nowadays it is not the serendipitous. First of all, we will just see the some history regarding the drug discovery, how this process is going to be started. If we will talk about pre 1919, that time these drugs were discovered from the herbal drugs or serendipitous studies. And in 1920s and 30s, we know that the discovery of penicillin, this era is known as the vitamins and the vaccines era. In those era, all the vitamins or vaccines are maximally discovered. In 1940s, during the World War II, and there is a need of new drugs for the treatment of
जिंदल
the.
सर
Hello.
स्लाइड ना चेंज नहीं हो रही है फर्स्ट स्लाइड पे ही टिका हुआ है
Okay, let me see.
हाँ एक बार देखिए आप साइड से स्लाइड चेंज कर लेना एक बार चेक कर लो अब स्क्रीन शेयर कर लो फुल स्क्रीन
वही कर रहा हूँ सर.
बस ठीक है
अब चेंज हो रही है सर.
यस
Okay. So, in the era of 1940s, during the World War II, lot of new diseases are there due to the war. So, new drugs were discovered in that time. But this drug discovery is on its own wings with the discovery of DNA in 1950s. In 1960s, this discovery process is more vast, and in the 1970s, rise of biotechnology and with the use of ITs, information technology, drug discovery becomes prominent. In 1980s, there is the commercializations of drug discovery was there. In 1990s, there is a robotics and automation process involved in drug discovery, and nowadays there is the involvement of artificial intelligence, machine learning into the discovery of the new drugs, which will very effectively help in the discovery of the new drugs. So these are the expenses incurred by the various top companies on the R&D in the discovery of the drugs. So it is so by showing this, I am just telling to you that all the top companies are very much uh, interesting to do this drug discovery for the new disease or existing disease, which is very important part of our research. So, if we will talk about what is drug discovery, how we can define, this is the process of identification of disease and therapeutic target. Basically, by in a drug discovery, we discover a new therapeutic moiety for a particular disease, and for that therapeutic moiety we search methodology, assay development, lead identification, in vitro characterization, formulation and animal pharmacological studies, pharmacokinetics, safety studies in animal. In the last we will do the clinical trials in the human one. So this is the whole process, as well as we can say that the definition of a drug discovery. Various stages of drug discovery include basic research, feasibility studies, program, non-clinical development. In the last, that is the clinical development. So this is a one timeline of a drug discovery development of any compound. It is not a very simple process, it is a tedious process, and it will involve around 15 years to develop a one drug. If you will search at 10,000 compound in earlier stages of drug discovery, it only 250 compounds were goes into the preclinical studies, and 5 compounds were goes into the clinical trial, and after the clinical trial only 1 or 2 compound get approved by FDA, and these compounds goes into the market for the consumption. So it is a very tedious as well as time consuming process of drug discovery. So, drug discovery, in this uh, process, target of interest are selected on the basis of their pharmacological basis. Just I told you earlier that first we have to select the disease. For that disease, we will search the target. So these are the various methods of drug discovery: random screening, molecular designing, drug metabolites, and serendipity. So these are the systematic process by which we will do the drug discovery. First, we will select the particular target. Then we will uh, go for the lead discovery. Then we will synthesize the that moiety by use of medicinal chemistry and our other sciences. Then we will do the various in vitro studies on that compound. Then we will go for the animal studies, which is known as the preclinical studies, and in the last we go for the clinical trials, and after that the drug goes into the market. So first, if we will talk about the first process, which is known as the target selection. Target selection is basically a drug discovery is defined as a decision to focus on the finding an agent with a particular biological action that is anticipated to have a therapeutic utility, which is the target. Target is that moiety for the, where the therapeutic utility is connected with that target. So first, our motto is to identify the target, identify molecular target that are involved in the disease progression. We have to find that target which is involved in the progression of the disease, and we will validate that target at the molecular level. So when we select the target, ki this is the target responsible for the disease, then we will go for the any drug or moiety which is going to target that particular selected target. For target selection, these are the various biochemical classes of the drug targets like G protein coupled receptors, various enzymes, hormones and factors, ion channels, nuclear receptors. And these are the following are the techniques of selecting the target. First is cellular and genetic target. These involve the identifications of the function of a potential therapeutic drug target and its role in the disease process. First of all, we will identify the potential therapeutic target and that whose role in the disease process. Then we go for the various other things like genomics. The study of genes and their function is known as the genomics. Genomics aims to understand the structure of genes, including the mapping genes and sequencing the DNA. Genomics जो है, it is a one of the way by which we can select the target based upon the genes. Then proteomics. This is the next way by which we can select the target. It is a study of proteome, the complete set of proteins produced by a species using the technologies of large scale proteins separation and identification. And around 90%, 91% of the drugs, our existing drugs are act at the proteins level. So this is the potential way to select the target. And we will analyze the proteins which will be the responsible for the that target, and and we will select that proteins for the as a target selection. So in the proteomics, by the use of proteomics, we will compare the expression level of the normal protein as comparison expression by comparing the protein expression levels of normal tissues with the diseased tissue. How the diseased tissues expressing their protein level and how the normal tissues expressing their proteins level. By comparing these, we will select the diseased tissue as the target one, and for that we will going for the selection of the various lead compound. And the last way of selecting the target is bioinformatics. It is a branch of molecular biology that involves the extensive analysis of biological data using computers for the purpose of enhancing the biological research. And bioinformatics having the very much vital role in the identification of the target as well as computer screening of chemical compounds. And bio with the use of bioinformatics, we compare the entire genome of pathogenic and non-pathogenic strains of microbes and identifies the genes and proteins associated with the pathogenism. Means to say that in diseased condition, we will identify the pathogenic genome, कि who is the pathogenic genome which is responsible for the disease, that is the target. And we will uh, do our whole work according to that target. So this is the first step of our drug discovery, that is the selection of target and optimization of that target. After that, the second process is lead discovery. What is lead? Lead is known as the compound which is the organic in nature, organic molecule that act as a prototype drug. First, we have think about this should be the compound which is a prototype drug, and after that we will optimize that compound with the continued research. We will identify the small molecules which is a function of any protein function is a response as a act as a lead compound. The process of transforming these into the high content lead series. So in the lead discovery, various approaches are there. First of first one approach is serendipity. For a lot of compounds are successfully discovered using this technique also. Example is levimisole and valproic acid. The second is way of discovering the lead compound is endogenous source. Human diseases are basically how these diseases are progress, how these diseases arises. These diseases are arises from the disturbances of normal biochemical processes inside our body. So the most appropriate approach is to we will do we will mimic the endogenous peptide activity with the non-peptide, and we will give those non-peptide inside our body, and we will see the activity of those mimicking non-peptide, and by this way we can also discover our lead compound. And third is exogenous source. Exogenous source out from the out sources like from the pharmacological sources, from botanical sources, from plants and animals. We will we search various compounds. We will extract various compounds from the plants and animals, and those compounds are responsible for the activity. And the last one is the rational drug design. By we are synthesizing various compound rationally, and checking those compound of how they are going to be target. This is the fourth way of discovering the lead compound. So first of all, for the lead discovery, synthesis and isolation. In this process, separation of mixture we will do separations of impurities. Various impurities are also responsible for act as a lead compound for any other disease. In vitro chemical synthesis is the process by which we can synthesize various organic molecules. Biosynthetic intermediate. In the biosynthetic pathways, various intermediate as act like a lead compound or organic molecule. So the second we can use the combinatorial chemistry. In this, that is the rapid synthesis or computer simulated of large number of different but structurally related molecules we will synthesize, and we will search a new leads and optimize the target affinity and selectivity by various processes. Then we will go for the ADME properties of that synthesized compound, and we will in the last we will reduce the toxicity and eliminate various side effect of that. This all comes under the optimization of that lead compound. Then high throughput screening. In this we are by use the computer, by use the database. In robotics, millions of compounds are to be tested on particular target in computer system is by using the software system, and we will screen the particular compounds which are responsible for the target that going to be those 20 to 30 compound which are responsible for curing that disease. Then we will synthesize only those 20 to 30 compound, and uh, do all the studies on those compound. By this we can eliminate the various synthetic chemistry process, जिससे कि हमारा बहुत ज़्यादा टाइम कंज्यूम हो जाता था पुराने टाइम पे उनको सिंथेसाइज करने के लिए। तो हमने इसको बहुत ज़्यादा स्ट्रीमलाइन कर दिया बाय यूजिंग दिस सॉफ्टवेयर टेक्निक्स। Then, when when we have selected one molecule lead, then we will go for its synthesis with the use of medicinal chemistry and or in this way we used various drug discovery process, lead optimization, heat to lead। पहले हिट्स कर रहे हैं ड्रग डिस्कवरी में, उसके बाद वो एग्जैक्टली वो कंपाउंड कैसे हमारा काम करेगा। फॉर दैट first of all, that is the development of library. In this we will collect collection of stored chemicals along with associated database. It will assist in high throughput screening and help in the screening of drug target, and all these processes are based upon the organic chemistry. Then we develop the SAR studies of that particularly lead compound. We will identify the pharmacore which is responsible for the that biological activity. It is the precise molecule which is the responsible for biological activity, and we will al- allow to eliminate the excessive functionality, and by this way we come to know about the structure activity relationship of that particularly lead compound. Then we will go for the in silico screening. This is again by using that computer software techniques. We go for the in silico studies. In the last, we go for the chemical synthesis of that particular selected compound. In this, that is the in we will produce we will the in large quantity by using the synthetic way, because we have to test all these the selected lead compound on to as well as in vitro as well as in vivo, and after that in clinical trials, so we have to synthesize on a large scale. So this is the last step of that this medicinal chemistry, and after that the next step of our discovery is the in vitro studies. In the in vitro studies, uh, it will use the component of organisms, that is the test tubes experiment. For example, cells derived from multicellular organisms, subcellular component, cellular as well as subcellular extract purified molecules. Means to say that first of all जो है हम क्या करते हैं इन विट्रो स्टडीज करते हैं, ताकि हमारा एनिमल्स से पहले हम उसकी सारी सेफ्टी, एफिनिटी, सिलेक्टिविटी और उसके मैकेनिज्म ऑफ एक्शन को पता कर लें कि वो कैसे काम करेगा. फॉर दिस वेरियस टेस्ट ट्यूब्स एक्सपेरिमेंट्स आर देयर, और बहुत सारे सेल्स जो हैं हम कई ऑर्गेनिज्म से हमें इन विट्रो मिल जाते हैं. सेल स्टडीज करके हम अपने केमिकल अपने लीड कंपाउंड को इन विट्रो टेस्ट कर लेते हैं, और उस टेस्ट करने से हमें ये पूरा का पूरा पता लग जाता है कि हमारी टॉक्सिसिटी लेवल क्या है, हमारी ड्रग की डोज कितनी होनी चाहिए, कितना हम यूज कर सकते हैं. तो देन वी विल गो फॉर द एनिमल स्टडीज. दैट इज द theoretical and practical aspect of drug development process, that is the animal studies. In animal studies we go for the first preclinical studies. In preclinical study the aim is to satisfy all the requirement uh, before a new compound is deemed to be ready to be tested फॉर द फर्स्ट टाइम इन ह्यूमन. मेन बेसिक क्या है कि ह्यूमन पे टेस्ट करने से पहले हम एनिमल पे टेस्ट करके हम इंश्योर हो जाएं कि हमारा जो डिस्कवर्ड कंपाउंड है वो अच्छे से काम करेगा. सो दैट्स व्हाई दिस preclinical study is falls into the basically four category. First is the pharmacological testing, second is preliminary toxicological testing, third is pharmacokinetic studies, that is known as the ADME studies of the drug, and the last one is chemical and pharmaceutical assessment to assess the feasibility of large scale synthesis and purification. So first, preclinical studies will give us the information about the metabolic profile of the drug, toxicity profile of the drug, and various drug interactions. So for this various we develop various disease models, and various animals were used for particular disease. For example, in for the psoriasis we will use imiq mode induced mice model. In this Valvasi mice were selected, we apply the imiq mode cream, and the psoriasis develop, and we will test our developed compound on that animal, and we will do various biochemical testing of that skin and the histopathology of skin. By this way, by we test our new compound on the various animal models, which and developing the animal models which are very much similar to the human disease caused to the human also. And we can predict human toxicity in 70% of the cases. Then behavioral studies in the using the animals, we also investigate the behavioral results of the various drugs. For example, despair based force swimming test, tail suspension test, various reward based tests, anxiety based test we will do. And functional imaging, in the animal studies we will do various functional imaging. By this we can detect or measuring the changes in metabolism, blood flow, regional chemical composition, and absorption processes. So tracers are used like MRI, CT scans we are using at animals levels. And after that, various ex vivo studies were also performed to check the performance of the new discovered compound. Experimentations on tissue is an artificial environment outside the organism. मतलब हम एक artificial environment develop करते हैं, उसके ऊपर हम पूरा का पूरा study करते हैं. All this process up to preclinical step तक करने के बाद we go for the clinical trial. In this process we apply to the FDA for the approval of the clinical trials. उनको हम पूरा का पूरा अपना ड्रग का फिजिकल केमिकल डाटा देते हैं, प्रोवाइड करते हैं, और साथ साथ में हम पूरा का पूरा उनको प्री क्लिनिकल डाटा देते हैं कि आवर डिवेल्प्ड कंपाउंड जो है वो बहुत अच्छे से इस डिजीज को टारगेट कर रहे हैं और इस डिजीज को ट्रीट कर रहा है. काइंडली गिव अस द परमिशन फॉर द कंडक्ट ऑफ क्लिनिकल ट्रायल. आफ्टर studying all those things by the FDA, they will approve us for the clinical trial. And our clinical trials are also जो है वो हमारी इंस्टीट्यूशनल एथिकल कमेटी की अप्रूवल से हमारे इंस्टीट्यूट में स्टार्ट होते हैं. सो क्लिनिकल ट्रायल्स जो है इट इज अ सेट ऑफ प्रोसीजर इन मेडिकल रिसर्च एंड ड्रग डेवलपमेंट टू स्टडी द सेफ्टी एंड एफिकेसी ऑफ अ न्यू drug. These are essential to get marketing approval from the regulatory authorities. Without clinical trial हमें किसी भी ड्रग की जो है अप्रूवल नहीं मिलेगी. And clinical trial will require up to 7 to 8 years, and it is divided into 4 phases: phase 1, phase 2, phase 3, phase 4. All we know that these phase very well. In the phase 1, basically first stage of testing in the human subject. Only we will take 20 to 50 healthy volunteer, and we will check only the toxicity, tolerated dose, and pharmacological dynamical studies in the phase 1. And in phase 2, this is known as the controlled clinical trial, and we will take 50 to 300 patients. It is known as a controlled single blind technique, and it is concerned with the main safety, efficacy, drug toxicity, and any drug interaction found जो है वो हमें फेज 2 में पता लग जाता है. Then in phase 3, it is extended clinical trial. In this, we take a more and more patient, from 250 to 1000 patient, and it is the most expensive and time consuming process. And it is in this we will use the double blind technique, and it is concerned with safety, efficacy, comparison with the other drugs package. And also we will develop all the writings information which we want to give to the normal public. So पैकेज इंसर्ट, जो एक किसी भी मेडिसिन के अंदर एक पैकेज इंसर्ट डाला होता है, उसके अंदर जो भी इंफॉर्मेशन है, वार्निंग इंफॉर्मेशन, ड्रग इंटरेक्शंस, all those were developed during these clinical trial. And the phase 4, which is known as the post marketing surveillance, once after the phase 3 we got the marketing permission, and the drug goes under the phase 4 trials in post marketing surveillance. This is designed to detect any rare or long term adverse effect, एडवर्स ड्रग रिएक्शन. ऐसा भी होता है बहुत बार फेज 4 के बाद, मार्केट में ड्रग आने के बाद हमारी ड्रग मार्केट से वापस बुला ली जाती है ड्यू टू वेरियस साइड इफेक्ट एंड अदर इफेक्ट्स. सो दिस इज the all the processes of drug discovery. The first we will summarize it. First we will go for the selection of the target, कि कौन सा टारगेट होगा जिससे डिजीज प्रोग्रेशन हो रहा है. उस पर्टिकुलर डिजीज के लिए, उस पर्टिकुलर टारगेट के लिए सेकंड हम एक लीड कंपाउंड डिस्कवर करते हैं बाय द सेकंड स्टेप. In the third step of using medicinal chemistry, we will extract or synthesize that compound. And in fourth step, in vitro, हम जो है उस पर्टिकुलर जो लीड कंपाउंड है उसको टेस्ट करते हैं कि वो हमारा पर्टिकुलर टारगेट के लिए कैसे काम करता है. एंड इन द फिफ्थ स्टेप we will do all the animal testing of that developed compound, and after that we will go for the clinical trial. This is the whole drug discovery process, and these are the some drugs which are recently approved by FDA in 23 for various that is the lecanemab drug for treatment of the Alzheimer's disease. That is a Branjavi drug to improve the glycemic control in the adult with the type 2 diabetes. And these all various drugs are approved by FDA in every year, which are for the new for the existing disease, also for the new diseases also. So thank you very much for the patience listening.
Thank you, sir. It was a very nice presentation, and you have broadly explained about the drug discovery starting from target selection to clinical trial phase 4. And I hope the population is going to be benefited with your valuable suggestion and talk. Thank you, sir. Thanks a lot.
Thank you, sir, for giving me the opportunity in this lecture series. It is my privilege to be a part of this lecture series.
Thank you, sir. Take care. Okay.
Okay, sir. Bye.